How did it get to be the end of May already? So one more week left. Next week we'll have giant viruses, guest lecture on virophage. If that doesn't go for a whole section, I might actually finally get to the influenza extra vaccine stuff that's been tucked at the end of every single lecture that people have been downloading for the last 2 or 3 weeks. And then review next Friday. A week from Tuesday, 8 a.m. is the final. I will be maybe available really briefly on Monday morning if people have extra questions in finals week. Otherwise, I will not be around. I'll be sitting in an airplane. It'll be so much fun. But at least we'll be available on email, at least for part of that day. Okay, so last time we talked about, move on, archaeal viruses. Again, this is something we could spend a whole term talking about, and maybe at some point I'll get around to actually doing a whole course on archaeal viruses. These are almost all double-stranded DNA viruses. So far, no one has found any RNA viruses that infect archaea. May have to do with the fact that mostly people are looking in extreme environments, but also there are very few RNA viruses that infect bacteria either. We talked about Q beta and MS2 very much at the beginning, but for the most part, viruses infecting the prokaryotes, bacteria and archaea, are double-stranded DNA viruses. The ones that infect Euryarchaea, for the most part, are bacterial-like, but again, there are always exceptions, and I think that archaeal viruses are exceptional, pun completely intended, mostly because of these current archaeal viruses that we work on. These are the thermophile, acidophile viruses that have these most amazing shapes and genomes and fascinating kinds of things to look at, and will hopefully keep me busy for the next 20-odd years. So any questions about that archaeal virus lecture? I know we skimmed over a lot of stuff. I'm not going to ask you little nitpicky details on exams like, how many nucleotides are there in the CIF1 genome. But I think some of the cool take-home stories like folded lipids and the strange shapes like the bottle-shaped virus, those are pretty reasonable things to ask. And the interesting ends of genomes, because as George pointed out in his guest lecture, Stedman just loves these comparative questions from different viruses and virus families. Okay, if there's nothing on that, we'll talk about retroviruses today. Again, this is sort of the end of the term, all the bizarre and different viruses that we haven't really talked about yet. Retroviruses are really interesting, mostly from the way that they replicate. Yes, we know a lot about retroviruses because of disease, but my big interest is really in how they are molecularly replicating, because they package, well, at least the standard retroviruses, package single-stranded RNA in their virions, but they have to go through a double-stranded DNA intermediate. And there are sort of similar kinds of reverse transcriptase requiring viruses. Some actually package DNA, some are packaging RNA, some are packaging single-stranded DNA. So these viruses and their virions really go through the whole process, RNA and DNA and back again. So really fascinating process. These were originally studied actually not because of HIV, but because these were RNA tumor viruses, and I promised to bring my tumor viruses books before. So this is what was known about retroviruses before HIV, that these were the RNA tumor viruses, and actually that helped a lot with understanding HIV and retroviruses, because people have been studying these tumor viruses and RNA tumor viruses. And I also promised the DNA tumor viruses, the papillomas and polyomas. This is, as you see, a considerably less thick book than the RNA tumor viruses. Well, these are, these I think from the early 80s, these textbooks. So the idea of RNA tumor viruses, and RNA tumor viruses immediately should be sort of something you go, well, wait a minute, what's going on with an RNA tumor virus? Because cancer is a genetic disease, genetics is DNA. How do you have an RNA virus that's causing a DNA disease? So that was immediately a question that people started to think about. Oh, there's something weird about what's going on with these quote-unquote RNA viruses. And yes, HIV, human immunodeficiency virus, mostly one. There's a few other ones as well, but most of the work's been done on HIV-1, and we'll spend a while talking about that. And then the Fullerian cone should be a wink, wink, nudge, nudge, similar kind of structure to something that we've talked about a little bit before. There are a bunch of key concepts as far as these retroviruses are concerned, mostly having to do with their replication. Again, my big interest is in the molecular aspects of these things. So one big difference as far as all of the other viruses that we've talked about so far, their primer for replication is a tRNA. And actually, it's a cellular tRNA. And we'll see how that works a little bit later on. We've talked about frame shifting quite a bit already, particularly for the coronaviruses. It's where when you're translating, so the ribosome is chugging along the RNA, when that then switches reading frames and gives you a different protein. That's also absolutely required for all of these retroviruses, including HIV. There's splicing, which is also required for the functions of these viruses. Reverse transcription, and the protein which does that, the reverse transcriptase. Reverse transcription is really pretty straightforward. Again, it takes RNA and makes it into DNA. The reverse transcriptase, on the other hand, is a really fascinating enzyme because it doesn't just take RNA and goes to DNA. It takes RNA to DNA, DNA to DNA as well. And also the reverse transcriptase has enzymatic activity in that it has an RNase H activity as well. So there are multiple different activities that reverse transcriptases have, and it's not just reverse transcription taking your RNA and going to DNA. Why do we have this reverse transcription? It seems to be, at least in the case of the standard retroviruses, all about integration and getting that RNA integrated into a DNA genome. How do you do that? Of course, it needs to be DNA before you can integrate it into the DNA genome. And it turns out that this process is very similar to what happens in Lambda and some other viruses that we haven't talked about. It's a pretty random integration, however, which is unlike what happens in Lambda. LTRs, yes, we needed more acronyms in this class, didn't we? LTR just stands for long terminal repeat, and that is one thing that comes out of the replication process. So not surprisingly, terminal repeats, identical sequences at either end. Also very important for transcriptional regulation, so the LTRs are really good promoters. Template switching, we've talked a little bit about template switching already in terms of the coronaviruses. This is where you have the polymerase, these coronaviruses, of course, RNA-dependent RNA polymerases, which can jump to different parts of their template, and that's how you get the nested subgenomic RNAs. In the case of these retroviruses, the jumping seems to be between different genomes or different ends of the genome, and we'll see how that happens as well. And then again, more terminology, GAG, Pol, ENV. This is what people always call the polyproteins which are made by these viruses. And immediately, polyprotein, you should be thinking, okay, we're going to be making one big protein or lots of big proteins that get chopped up by proteases. So again, very similar to some of the picoRNA viruses, the flaviviruses, et cetera, that we've talked about before. So in general, our outline again, origins and disease, a little bit by structure and entry, but the big deal here is replication and how you go from a single-stranded RNA to double-stranded DNA and back to RNA again. And then, then the sort of standard stuff that we have here. Where do you find retroviruses? Before we got to all these immunodeficiency viruses down here, the lentiviruses, these were all about cancer, sarcoma, tumor virus, leukemia, sarcoma, etc. So all of these were the RNA tumor viruses, and that's what people were studying before HIV came along. And we'll talk about sort of why that is a little bit later on. All of these viruses, again, are completely dependent on reverse transcriptase. The foamy viruses down here at the bottom, these were only found after all of the other ones were, and these are a really nice example of a retrovirus that doesn't seem to cause any kind of disease whatsoever, and is a really nice model system for studying retroviruses, of course, that are not causing disease, if you want to study a lot of the general molecular basis for how a lot of these things work. But in many cases, these tumor viruses, again, were the ones that were being studied. And in fact, it was the study of these viruses that oncogenes were originally found. It was studying literally Rous sarcoma virus, where they discovered what the oncogenes were, and then eventually figured out that oncogenes were actually genes that were normally present in the genome, and that these viruses had picked up these genes in order to be causing cancer. A standard retrovirus, virion, looks a lot like this. These have lots and lots of structural proteins that are associated with them. Unfortunately, the terminology, particularly for HIV, is ridiculously convoluted, so I'll try and keep track of these things. But they're envelope viruses with an internal capsid. In the envelope, there's a surface protein that sits on top of a transmembrane protein, and these guys then have a matrix protein underneath them, basically interacting between, again, what's on the outside and what's on the inside. This matrix protein, here it's shown as a gap, but it also interacts directly with the capsid proteins. The capsid proteins are going to encode your genome, what's being packaged. This now is really quite different, again, than any of the other things that we've talked about before. We always talk about the retroviruses having a positive strand RNA genome. They've actually got 2 copies. All of them have 2 copies of the positive strand RNA, which is packaged. And then attached to those by base pairing interactions are cellular tRNAs. So now this is an example of cellular nucleic acids that have to be packaged as part of the genome, or sorry, part of the virion, I should say. So this is, again, a big difference, and I've talked about them before. They're the primers that you need for replication. But if you don't have cellular tRNAs packaged in your virion, you're not going to get any kind of replication taking place. These RNAs, and it's a positive strand RNA, so at least in theory, once it gets inside the cell, it can be translated and made into protein. Turns out that that's not the case, and we'll see why that is a little bit later on. It actually has to be reverse transcribed, and that reverse transcription has to give you a double-stranded DNA that gets inserted into the viral genome before any viral proteins are made. And so these are actually kind of like what you see in the negative strand RNA viruses, where you have to have an RNA-dependent RNA polymerase in the virion. Here, you have to have the reverse transcriptase in the virion as well. So the reverse transcriptase is present in the virion together with the RNA. You also have the viral integrase, again, an enzyme present in the virion, because the double-stranded DNA just by itself can't do anything. It's got to be inserted into the genome before it can actually be functional. So you need these enzymes as well. There's one more enzyme which is also always present in the virion. That's a viral protease. Now, why the heck would you have a viral protease here? It turns out that the viral protease is just left over from the maturation step that happens at the very end of virion formation. So lots and lots of proteins in here, and as we'll see, there's actually one more structural protein that you have in HIV that's a little bit different. But let's focus in on the genomes now and see what these genomes look like. Genomes have normal caps, normal tails, so at least, again, in theory, this could be translated. And for reasons that are not completely clear, but probably just because it's in the capsid itself, they don't get translated. Reverse transcription happens first. However, between the cap and the tail, you've got a couple of interesting sequences. First, this R sequence. R just stands for repeat. So it's a directly repeated sequence. It's identical at both the 5' end and the 3' end of the genome. Then you have unique sequences at the 5' end and unique sequences at the 3' end, next to a primer binding site. And then, as we'll see, this polypurine tract over here, this also serves as a primer for later on in replication. Then we have the proteins. GAG, POL, and ENV. GAG stands for group-specific antigen. That's unfortunate, again, from the studies that came out of this when people were studying the various different RNA tumor viruses. So group-specific antigens, but antigens, that means these are proteins that are being made in large amounts because you're getting antibodies to them. And so these are your major structural proteins, particularly the parts that are in the inside of the virion. The matrix protein, the nucleocapsid protein, and the capsid protein. Nucleocapsid I didn't mention before, but that's just binding to the genome. Again, one of these pretty standard things that we've talked about before. The envelope proteins over here, ENV, surface protein and transmembrane protein, and then the POL proteins, which unfortunately also include the protease and the integrase, but these are where all the enzymes are. So enzymes, internal structural proteins, external structural proteins. The only other things I haven't talked about on here are the 5' splice site, or the major 5' splice site, major 3' splice site, and this Psi sequence, which is the viral packaging sequence. So this is the genome, and now let's look at a cartoon on how retroviruses get inside of cells. They bind to receptors on the plasma membrane, and these receptors are very, very specific. So a particular retrovirus is going to only be able to infect a very specific cell type. So for HIV, it's T cells, CD4 positive T cells, but doesn't infect other cell types at all. So binds to a specific receptor, fuses at the plasma membrane, but again, there's this capsid on the inside. So you release the capsid inside the cytoplasm. In the cytoplasm, now you have the availability of nucleotide triphosphates, and particularly some DNTPs. These then start the reverse transcription process. So virions, when they come inside the cell, actually are active. They are going to start making the DNA from the RNA. And this was, in fact, how reverse transcriptase was originally found. People looked at retroviral virions, threw labeled DNA at them, and saw that they incorporated labeled DNA. And this is why Howard Temin and David Baltimore ended up getting the Nobel Prize for discovering this new process of going from RNA to DNA. So once you have replication of the genome, and we'll look at the actual processes of replication here in much more detail in just a second, then you end up with a double-stranded DNA with ends that are associated with the integrase, and then this whole complex, so DNA plus integrase, needs to get into the nucleus. And there are sort of two processes by how this happens. The normal retroviruses actually just wait until the cell divides, and then they get into the nucleus. The lentiviruses, like HIV, actually have an extra protein that helps import that genome segment, which is a double-stranded DNA form of the genome, into the nucleus. Once it gets into the nucleus, there's integration. That's this blue thing right here. That then gets transcribed by cellular machinery, so cellular RNA polymerase. It will make various RNAs, some of which are spliced, some of which are less spliced, and lots of different splicing that takes place. The non-spliced form, you end up with lots of this GAG protein. Again, this is the structural inside stuff, matrix, capsid, nucleocapsid. Occasionally you have frame shifting and make some of the polymerases as well. Again, these are all now polyproteins. You have some splicing that takes place between that 5' splice site and 3' splice site. These then are your transmembrane and surface proteins. These get exported to the Golgi, endoplasmic reticulum, and end up making surface proteins that end up in the surface. Eventually, you make genomic RNA. That gets exported out to the surface of the cell, and you get packaging that takes place here. Packaging for retroviruses is also interesting because it's incomplete. The packaging that happens in these retroviruses, you still have to have more changes that happen to the retroviral proteins before they're actually infectious. So this last step down here is what's called maturation. This is where you have the final proteolysis steps that take place. So talked about proteolysis a bunch of times already. It's sort of providing that last priming step, so you actually end up with an infectious particle. So here, the proteases, and the protease is then just still in the virion, as we mentioned before, just because it's doing this process right at the end. So let's zoom in on genome replication. This is I think someone called it the Baroque mechanism for genome replication for these retroviruses. Really fascinating. So we already mentioned that when the genome comes inside the cell, it has this tRNA that is base paired to the primer binding site. And the tRNA is the primer for reverse transcriptase. So here, as always with all DNA polymerases, reverse transcriptase goes from 5' to 3'. So adds to the 3'OH, this is the end of the tRNA, and will copy the RNA out here to the end of the genome. This is this orange piece right here. Gives you the complement of the unique sequences at the 5' end and this repeat sequence, again, which is present at the 5' end and the 3' end of the genome. When the reverse transcriptase gets to the end, it stops because there's no more template. Then it also has RNase H, and again, the reverse transcriptase itself has RNase H activity. One of the domains of the reverse transcriptase has an RNase H domain. So it chews up the DNA in the RNA, chews up the RNA, because it's RNase. RNase H chews up the RNA in the DNA-RNA hybrid. Now you have a repeat complement and complement of the U5 sequence. Well, what do you have at the other end of the genome but a repeat sequence? So now this piece right here can loop around and base pair over here. Once it's looped around and base paired over here, now we're in great shape. This can now extend all the way out to the other end of the genome, where you have your primer binding site. This whole piece now is DNA. Again, the orange is DNA. This RNA now starts to get chopped up. And the last little piece that's left is this polypurine tract. And polypurines just apparently are resistant to getting degraded by RNase H. So this provides the primer for your second strand. Because all of this is negative strand DNA. This is a positive strand RNA virus. That negative strand can serve as the template once all of this has been degraded. That gives you positive strand, and people also talk about this as a plus strand, going from this polypurine tract, again, out through the end of the genome, and also copies part of the tRNA. Now, this part of the tRNA right here, that's what was binding over here to the primer binding site. You still have more RNase H. You chop up the tRNA. You chop up the polypurine tract. Now you have this primer binding site that can base pair over here to this primer binding site. Now you have a piece over here that gets filled in and the rest of the positive strand that gets filled in. So what this generates is a double-stranded DNA that's slightly longer than the RNA that we started with here in the first place. It's given you what used to be unique sequences at the 3' end are now at the 5' end. And the unique sequences that were at the 5' end are now also at the 3' end. This set of sequences, the unite sequences at 3 together with the repeat sequences and U5, is the long terminal repeat. So that long terminal repeat is only generated after you have replication has taken place of your genome. This process, reverse transcriptase, probably many of you have heard about. HIV mutates all the time. The reverse transcriptase has no proofreading activity. So as it's making DNA copies, it inserts lots of mistakes. So it's a low fidelity reverse transcriptase. And what that means is you have a very large number of slightly different genomes present in any cell which is replicating one of these retroviruses. It's true for the RNA viruses as well. But what this generates is something what's called a quasi-species. So there's no sort of one sequence, which is exactly the sequence which you have in any given infected cell. It's always going to be a mixture of different species. And that's very important for selection purposes, but also for drug resistance, as we'll see a little bit later on when we talk about therapy, particularly for HIV. And there are questions on this replication process, because this is kind of a weird and wonderful, yeah.
So if it's always mutating itself, what is the percentage of viable mutations to non-viable mutations?
Okay, so the question is basically, if it's mutating all the time, isn't it sort of killing itself, which is still paraphrasing? Yeah.
Just making a bunch of extra junk and then hoping
Right.
that
Every now and then something actually works. So one of the things that I think I mentioned way back at the beginning when we were talking about particles to PFU ratio, so number of infectious particles versus number of genomes or particles in general, HIV is one of the lowest of that. So 1 in 1,000 very often are actually infectious. But since it makes so many of them, doesn't seem to be too much of a problem. Yeah, what's that?
I think it's along the same line of if it's a pseudospecies, how often is it becoming a full species? Or, like, you see what I'm saying?
Yeah, so what do we, to some extent, what's sort of a definition of the quasi-species, maybe is a better way of putting that, if I may. So the idea of the quasi-species is you have lots of different, but not that different. There'll be a couple of nucleotide changes in your genome at any given time. And so you're going to have, basically what it says is you're not going to have a set genome and call that your species. It's going to be, people think of these as clouds of sequences which are all very similar to each other. Many of them have deleterious mutations, but enough of them don't. And what happens is that really provides an area for selection. And so if there's some kind of, again, I wasn't going to talk about this now, but we can talk about it now. If you have drugs that you're trying to use to treat something, as we'll see later, a lot of the drugs actually target these reverse transcriptases because you don't have reverse transcriptases inside your cell, at least not those that are similar to this. But if those are drugs that are binding to a very specific place in your DNA-dependent RNA polymerase, the reverse transcriptase, then any kind of mutation then will be resistant to those particular drugs, and then those, under a selection process, will end up dominating your population. And so basically what it does is it gives you a lot more genetic diversity to evolve with. So these are evolving very fast, but mostly because they've got a huge amount of diversity that they can be using. That's sort of the idea. Yeah, Mika?
I don't know if you already mentioned this, but I was just wondering, what is the difference then between quasi-species and HIV1 and HIV2?
Okay, question is, what's the difference between quasi-species and HIV1 and HIV2? So HIV1 and HIV2 have much larger sequence differences. If I remember correctly, and I may well not, I think it's about 20% sequence difference between the two, so it's pretty large. Whereas in quasi-species, each of the individual genomes will maybe have 0.1, 0.2% difference between them, but there are lots and lots and lots of them. Okay, so the thing that does this job is this amazing enzyme, reverse transcriptase. Inside virions, you've got lots and lots of copies of the reverse transcriptase enzyme, between 50 and 100. These, again, as I mentioned before, they're functional in virion preparation. So as soon as you get a virion that's got access to dNTPs, it will start to make DNA copies. It's got 4 different activities. The RNA-dependent DNA polymerase, this is your classic function, take RNA, make a DNA copy. But it's also a DNA-dependent DNA polymerase because it's making that second strand. It's still the reverse transcriptase, which is taking a DNA copy and making or DNA strand, I should say, and making a second DNA strand associated with it. I already talked about the RNase H domain, but it turns out that reverse transcriptases also have DNA helicase activity. And so they can pull 2 strands apart as well, and this is particularly important while they're replicating. So it always shows in that last figure, RNase H has degraded all of the RNA, which is a little bit of RNA which is left, and the reverse transcriptase can just go through and take that out. The RNase H, this is again a domain of the reverse transcriptase we'll take a look at on the next slide. Both endo and exo DNA and RNA hybrids. So it'll cut in a DNA-RNA hybrid right in the middle, your endonuclease activity, but also chew in at ends. And then also the DNA helicase activity helps to get rid of this. So this is the structure. Obviously, you can tell exactly that it's got this hand structure, very similar to RNA-dependent RNA polymerases and also DNA-dependent DNA polymerases. The big difference here is it's got this RNase H domain that's hanging off of the back of it here. These are also dimeric proteins. A little hard to see here, but there's a gray in the background. So gray versus colored. They're dimeric proteins. One of the dimers has the RNase H domain. The other one doesn't. But they're just binding right next to each other, and this is what's going to give you all of that replication. One of the things that you may or may not have guessed from this, DNA polymerases are by themselves really non-processive. They fall off all the time. So that's one of the reasons that it seems you've got so many of these reverse transcriptases in the virion. In theory, if it was processive, you just need two, one for each of the genome segments. But in this case, since they're not processive and they're coming on and off all the time, you actually need lots of them. And so where you have that positive strand where extend out to the end, or the negative strand extending out to the end, that's the major thing that you see, but you'll also have lots of smaller pieces of RNA in the genome that will also serve as templates. So actually it's not just the one polymerase going along or one reverse transcriptase, it's multiple ones making your genome all simultaneously. So with this, you've got, again, a double-stranded DNA genome. What do we need to do with it next? It needs to be integrated into the host. This is now your double-stranded DNA with the unique sequences, U3RU5, U3RU5, the long-term repeats at either end of the genome. The viral integrase binds to these sequences and then randomly integrates into the cell. Now, random in quotes here. Randomness, it seems to be actually have much more to do with the structure of the DNA that it's getting inserted into rather than the sequence of the DNA which is getting inserted into. So heterochromatin, way back when, molecular biology, really compacted DNA, very rarely do you have retroviruses that integrate into heterochromatin. However, euchromatin they do insert into. And this is probably why, as we'll see in just a second, it ends up often getting oncogenes, because they're actively transcribed regions of the genome. And this can actually be a problem if you're trying to use these kinds of viruses for gene therapy purposes. You pop them into an actively transcribed part of the genome, they might actually be in some kind of coding gene. So this random is all a bit of a, not completely random. We always say it's random. And those of you in the mutant viruses from hell class, how random our transposon insertions are probably also a bit of an interesting issue. But the integrase will, again, mostly binds to the ends of the genome here, makes a couple of exonuclease cleavages, cuts a couple of nucleotides off of the very end of the genome. This doesn't matter because, again, you've got repeated sequences at either end, so you're not going to need those later on. And then we'll cut offset in the genome of the host. It's not this particular sequence, but you end up with a gap. And so there's your viral genome. And again, this is a proviral genome. It's going to be sitting in the host genome, like the prophage that you see with lambda. And these gap sequences that you have at the end, this is just what happens with a lot of the transposons, again, that we talked about in molecular biology last term. You end up with a gap. This gap then gets filled in by the cellular polymerases, and that cellular polymerase will, in the process of filling in this gap, generate a directly repeated sequence. So here, GGGCC, over here, GGGCC. That's because that's where it cut up here and gave you the ends. And so it's a gap, just a single strand here because the double-stranded piece gets attached at the end. That gets filled in, so you end up with a direct repeat of genomic DNA, a direct repeat of your viral DNA, your virus genome, etc. So if you're just looking in genomes, which is really easy to sequence these days, and you see direct repeats followed by long terminal repeats, that gives you a really good indication that there's a virus, or there was a virus genome that was inserted there. We'll talk about these in just a minute, but this is where a lot of the human endogenous retroviruses were discovered, literally just in genome sequences, because you could see long terminal repeat sequences, and you had directly repeated sequences on either side of them. And so it tells you that there was this integration event that had taken place. Yeah, Melissa. Yes, so this video is really good for integration. It's not very good for the replication process. I'm still looking. If anybody can find a really nice animation of the replication, I would love to see it. I've tried to put one together myself. I'm horrible at animation, so it never came out very well. But that process I would really like to see. There probably is one somewhere. I just have not been able to find it yet. If we've got time at the end, I'll show what I think is a really, really nice video. And if we don't, you're welcome to look at it later, where they go through the whole replication process, again, with the exception of the genome replication. Okay, so I'm done answering questions. Are you ready to answer questions? Again, we're aiming for maybe all 100% all the time today. So, I need my clicker thing here. No, we can just start it now. The primer for the positive strand of retrovirus replication is a tRNA, a protein, the polypurine tract, a cellular primer, the genome itself. And I guess we're counting down from 3 minutes. Okay. 10, 5, 2, 1, stop. We have 76%. We want to call that good, or we want to talk to our neighbors? Let's talk to our neighbors. 30 seconds, go. Yeah, the positive DNA strand. I hear people asking questions about that. So by making the positive strand of DNA. Okay, things are getting quiet. Let's try again. How it ended up doing 3 minutes, 3 minutes and 10 seconds, I have absolutely no idea. We'll count down to 2 minutes. So again, the positive strand of DNA in retrovirus replication. I'm not going to tell you that. We have a few fewer of the outliers, but yeah, that's about it. Yeah.
So
10.
D is the cellular primer.
Mm-hmm.
tRNA be cellular, can it be a cellular protein?
I would say made by the cell. So, but in this case, more like a cellular regular RNA primer, like what DNA polymerases would use. Okay, anyone want to change their answer based on that? No, it'll be like D anyway. So, what do we think? We think about split between the tRNA and the PPT. So let's just step back here and look at this. So move this really briefly. So which is the positive strand? So the RNA, the RNA is positive strand, right? So that's going to be the DNA positive strand. It's going to be the top one, because it's going to be the same as that sequence. Right? Yes? No? The negative strand is synthesized first, but the positive strand is synthesized second. And so that's the one that comes from the PPT. So here it would be step 6. Synthesis of plus strand strong stop DNA. Happy, not happy? Probably could have been better answers to the questions. Make sure they're better proofread when they're on the final. Yeah, so the answer is C, not A. Okay, we talked about replication, now transcription. How does transcription work? Mentioned the LTRs, kind of beating this over the head. LTRs, the long-term repeats, these are really good promoters. Actually, they're some of the best RNA polymerase II promoters that anyone has actually analyzed. They are great binding sites for RNA polymerase II. Sits down, Tata box, B recognition element, etc. Transcribes the whole proviral DNA. The cap is regular cellular capping, regular poly-A tails, making this polyprotein either GAG and Pol or through splicing to give you the envelope proteins. And both of these are now polyproteins. They will get chewed up a little bit later on. And you have this RNA getting translated. Most of the time, it's going to stop translation right here after the GAG polyprotein, but in about 5% of the time, it reads through and will give you the Pol polyprotein as well. How does this happen? Actually, 2 different ways. There's either a frame shift that takes place in some retroviruses, and in other cases, it's just not reading the translational termination codon as a termination codon. Here's the example of frame shifting. Here we have the ribosome chugging its way along the messenger RNA, hits a secondary structure. There are always secondary structures that are involved in both translational suppression and in frame shifting. This is the normal stop codon that would be here. So if there wasn't any frame shifting, you end up stopping. If, however, and again, about 5% of the time, this ribosome bounces back one nucleotide. Now you don't have this UAA stop codon, you have a CAA, and you continue to read through and make all of the GAG-PAL polyprotein. And again, it's about 5% of the time that you end up with GAG-PAL altogether as opposed to just the GAG proteins. How do you finally put all of this together to make a virion? Proteases. So we talked about proteases again ad nauseum with the picoRNA viruses, the coronaviruses. Any one of these proteins that has a polyprotein that's associated with it gets chewed up into smaller pieces. The envelope protein gets chopped into TM and SU. The GAG protein, matrix, nucleocapsid, and capsid. And the PAL protein, protease, reverse transcriptase, and integrase. This transmembrane protein gets localized normally, because again, transmembrane is being made in the endoplasmic reticulum, standard membrane proteins. Getting the GAG precursor to the membrane, because it's the matrix, going to be assembling at the plasma membrane. There's a modification that happens. It's a small fatty acid myristate that gets added to the GAG protein, and that then will lead to this GAG protein being localized to the plasma membrane. And then you have to package genomes. So your genome packaging happens through this Psi sequence. This Psi sequence is also known as the packaging sequence. That's what the nucleocapsid binds to. Nucleocapsid will bind to the Psi sequence. And because this Psi sequence is here, it's between the 5' splice site and the 3' splice site. I'll have to work on pronouncing that. 3' splice site. Then you're only going to be packaging unspliced RNA. And that's important. You don't want to be packaging spliced RNA because that's not going to be functional in terms of a virus genome. So that's how that happens. And then again, the protease is active at the very end of your whole process. So that's the big picture for these retroviruses. There are a couple of other things that I wanted to mention. The first of these is, again, how these retroviruses were discovered in the first place. And these were the RNA tumor viruses. RNA tumor viruses look a lot like normal retroviruses, except they have this extra gene that's associated with them. And that extra gene is an oncogene. How do they get these genes? Well, what seems to happen is when you have that integration process, they integrate again into active parts of the genome. And so those are being actively transcribed genes. Though you have an integration of a retrovirus into that region, in some cases it ends up picking up a little bit of extra DNA. And that little bit of extra DNA, in a few cases, turns out to be some of these cellular proteins which, when they're being expressed, end up giving you cancerous growth. And this was how oncogenes, again, were originally found. People thought they were just the tumor viruses, but it turns out that they're now dysregulated because now, where the Src gene was before, it was actually being regulated normally inside the cell. Now it's part of one of these retroviruses. So this will integrate wherever it is in the genome. It's next to one of these LTRs, which is a gangbuster promoter. So it's going to be made all the time in the wrong place and leads to cancerous development. So that's where these guys came from. The other thing that we find, as I mentioned before, and looking through genomes, large portions of our genome contain these either endogenous proviruses, so they've got down here LTRs, GAG, PAL, and ENV, and another LTR together with these direct repeat sequences on the other side. There are hundreds to thousands of copies of these in our genome. We don't actually know what they do. People have actually recreated them, tried to see what kind of activity they have. Not at all obvious, but lots of them present in our genome. And there are also quite a few that are missing some extra proteins. And so the retrotransposons have GAG proteins, they have PAL proteins, but they don't have envelope proteins. Otherwise look identical to what would be a normal retrovirus being packaged inside the genome. And between the two of these, that's 8% of our genome. And that's almost, let's see, 6 times more sequence than of the unique protein coding sequences that make us human. So I like to think we're more viral than we are human. People argue, and I'm one of those, that some of the other elements in the genome, line elements and sign elements, that also contain reverse transcriptase genes, they may be derived from viruses. Again, this is highly controversial, but that would make us over 40% viral rather than just 8% viral. I'm happy to be 8% viral, but maybe 40% is even more appropriate. So should we do another clicker question and hopefully people do better on this one? Why are only complete genomes, hang on, let me start this, packaged in retroviruses? Due to frame shifting, absence of oncogenes, packaged together with cellular tRNA, 2 copies of the genome have to pair, or the packaging site is between splice sites. Okay, we've got even less of a consensus here, so I think it is something we're going to need to want to discuss. So go ahead, talk to your neighbors. If in doubt, guess. That's what, 5 choices, 20% chance? Stedman's standard answer. Oh, yeah. And it was C last time, that's true. So. Okay, you can keep chatting. Let's go again. Somebody's clicking all the buttons like back and forth that I can't tell from this. Ten, 5, I like E as well. The size site is between the 5' splice site and the splice site that I can't pronounce. So E is the answer here. It looks as if we're not going to have a chance to look at the great video animation at the end, but I do highly recommend people taking a look at that afterwards. And it's a great review, actually, for what we'll talk about here. So finish up talking a little bit about HIV. I could, again, spend hours talking about this, and there are whole classes about HIV. Big problem, but we have actually, I think, kind of turned the corner in many cases with HIV, which is really impressive. About 37 million worldwide are infected. Many of these are children, many of them in the developing world, mostly through childbirth and breastfeeding. Seems to be the main reason that a lot of, at least the kids, are getting infected. 75% know that they're infected. It means 25% don't. And that is really, I think, the major issue right now with HIV, because there are, as we'll hopefully have a chance to talk about, even if we don't, they're really good drugs for keeping viral levels low. And so if there's any possibility, please get yourself tested. And that's also true in the US as well. It's, if anything, not quite 25%, but still above, I think, about 15, 16% of people who don't know if they're infected. And again, they're great drugs that work for this. Yeah, about 1 million, and still 10,000 deaths a year in the US. So, important, interesting things. Okay, what about the molecular biology? The only difference here is VPR, this virion protein R for HIV. All these other guys are the same. They just have ridiculously named GPs instead. The capsid is extremely similar, except that instead of a icosahedral one, you have this fullerene cone structure present here. And also you have, and it's actually, I don't have it shown on here, yeah, so VPR. So VPR is that extra protein that the other retroviruses don't have. Otherwise, this is identical to what we've been talking about before. This is that wonderful fullerene cone structure, which might, if you squint at it a lot, look a little bit like our friends SSVs in terms of how they're fitting together, but that's the only take-home message there. The HIV-1 genome is very similar to the other retroviral genomes because you've got GAG and POL and ENV, but there are a number of accessory proteins in the HIV-1 genome, many of them overlapping other reading frames, and they're all about this differential splicing. So differential splicing that takes place giving you a lot of these accessory proteins. But otherwise, LTRs, the way that these replicate is basically identical to everything that we've talked about in terms of the other retroviruses. How they get into cells, as I mentioned before, the HIV recognizes T cells, which are a really important component of the immune system. That's why HIV causes AIDS, because you're basically taking out the immune system because the virus is taking out all of these T cells. The surface protein, I'll call it, also GP120, binds to CD4, which is present on many, many T cells. And then the only way you can actually get HIV infection is if there's a co-receptor, CCR5. CCR5 is also involved as a cell surface, presumably transporter protein. Some people don't have CCR5. It's not clear why they don't have CCR5, but they're actually resistant to being infected by HIV. Yeah, Mika? Yeah, so Delta 32 is one of the variants that you have lacking CCR5. And so the 2 cases of cures have actually been bone marrow transplants from people who had deletions in CCR5. But doing bone marrow transplants is not a really good way for treating HIV. Then, once this gets inside the cell, you have reverse transcription that happens just the same. You have the integrase that binds to the end of the genomes. But now VPR allows this indosome to actually get into the nucleus before you have cell division. So that's, again, big difference between HIV and these other ones. The other non-structural proteins that are important for HIV, infectivity factors, unique factors. We're mostly going to talk about these 2, TAT and REV. TAT, which is a transactivator of transcription, and regulator of expression of virion proteins, REV. These are mostly important for regulation of RNA and RNA transport going in and out of the nucleus. We're going to skip this clicker question because I wanted to talk about TAT and REV. When the HIV genome is being transcribed, it makes this RNA that forms a secondary structure. This secondary structure, if you don't have the TAT protein around, this causes the RNA polymerase to terminate. So it actually doesn't make the whole messenger RNA, doesn't make envelope proteins, doesn't make GAG, doesn't make Pol at all. So just sits here and stops, terminates, etc. However, if there is the TAT protein around, that now binds to this RNA structure and basically serves kind of as an anti-terminator. So we talked about anti-terminators lambda before. Again, very similar process. The TAT binding to a secondary structure in the RNA causes the RNA polymerase to anti-terminate and transcribe the whole messenger RNA. And that, of course, is also going to be your whole genome, leads to production of the virus. So a lot of times when people talk about latent HIV, a lot of latent HIV, there'll be a little bit of transcription, but no TAT, so you don't end up making whole genomes. The other accessory protein that I wanted to emphasize is the REV protein. This REV protein serves as a shuttle for getting RNA in and out of the nucleus. So again, we've got lots of splicing that takes place in the RNA that's being used to make the HIV genome. Each of these spliced RNAs needs to get exported into the cytoplasm, gets translated, that protein needs to come back in, and multiple rounds here that have to take place of RNAs in and out of the nucleus. So this should be really similar to what we were talking about for flu. Again, where you're shuttling RNAs in and out of the nucleus all the time. This REV protein is involved in shuttling the RNAs in and out of the nucleus, and particularly important for getting the unspliced RNAs out of the nucleus when you need to package them. Because normally you have to have all your splicing and then all the splice junction intermediates, etc., the quality control that the cell has for RNA splicing. This now has been subverted because you want to get your whole RNA out and get it packaged. Again, unspliced RNA, usually unspliced RNA, bad news. So REV helps that process in terms of getting things out. Other HIV-specific proteins, I'm not going to talk about them. They won't be on an exam. I wanted to talk about HIV therapies. Again, they're really good. The first round of therapies were single therapies. This turned out to be a real problem because of the quasi-species and selection. But now if you use a combination, and I think the current antiretroviral therapies are 3 or 4 different drugs, they address the integrase. They're inhibitors of reverse transcription. They actually are also using some of these fusion and entry inhibitors. You remember way back when we talked about entry, the conformational change that happens in the fusion protein. There are now drugs that can address that. And there are protease inhibitors. So protease chopping up all of those pieces in order to get the final function available, that works. So according to UNAIDS, 22 million of those people who are HIV positive are getting these kinds of treatments. Deaths are down about 50%, still 2 million a year, but they've come way down. And there's one other thing that one can do. If you are in a situation where it's likely that you will be exposed, you have an HIV positive partner, for instance, you're an HIV negative, pre-exposure prophylaxis, you take reverse transcriptase inhibitors. These are nucleotide inhibitors, and basically what they do is they bind to the reverse transcriptase, but they cause chain termination because they don't have extra OHs, which can be extended. So, and this works extremely well, 90+% effectivity. So if you're exposed, A, get checked, find out your status, and B, either be treated if you're positive, or if you're going to be exposed, make sure you take PrEP. So we'll stop there. There's more scary statistics about in the US where people aren't getting tested, but we'll move on from there. Everybody enjoy what should be a really nice weekend, and we'll see you on Monday to talk giant viruses. Oh, and very last thing, I do highly recommend this video here that I linked. It's a great review for the whole molecular processes.